innovative new drugs, proudly hosted by the Department of Histology, Faculty of Medicine, Universitas Indonesia. My name is Priska, and I will be the Master of Ceremony of today's event. Before we begin, allow me to present the agenda for today's event. The first one is opening remarks. The second is introduction of moderator and speakers, followed by guest lecture and discussion, and the last is closing and group photo. Without further ado, let us proceed with the opening remarks. To begin, we would like to invite Ibu Dr. Ani Wardani Hakim, SSI Apoteker and Biomed, to deliver the first opening remark as Secretary of Master's Program in Biomedical Science. Bu Rani, the floor is yours.
Okay, good morning. Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like to say thank you to Histology Department and Stem Cell Cluster to make this event today. And then I would like to say welcome to Professor Alexander to our place. Thank you very much. You come here and then you will share your knowledge about animal model and cancer. Actually, our study program have about 160 students, and we have also concentration in histology and stem cell, and also microbiology and immunology that I think it is your expertise. So, and then the research nowadays, many students research about cancer, and they're doing it in animal model, and then they usually test about immune parameter like inflammatory markers and etc. So I think your topic, your sharing knowledge will be very fruitful for our students, of course us as a teacher. And then these students absorb the knowledge, and Professor Alexander, once more I say thank you very much.
Next, we invite the Secretary of Doctoral Program in Biomedical Sciences FKUI, Dr. Robiatul Adawiyah, and Biomed SP Park, Subspesialis Miko Konsultan. Dr. Ada, the floor is yours.
Thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Good morning for all. Thank you very much for coming to this event. And also thank you very much to Professor Alexander McLellan and also Prof. Jeanne for guest lecture today. I think in this topic, cancer animal models for in vivo testing of innovative new drugs, I think this topic is very useful for our study for our students in PhD program, because some of PhD program students take this topic in their dissertation and or their research. I hope our students could get a lot of things from this guest lecture. And also, I think one thing that I want to ask you, Professor Alexander, your expert is microbiology, right? I think I will. What about microbiology? Viral, bacteria, or also fungi?
I trained in bacteria.
Bacteria.
For my master's.
Okay.
But then I moved to immunology.
Okay.
For my PhD.
Oh, okay. Thank you. Actually, if you are also expert in fungi, I will talk a lot about.
I eat mushrooms.
Yeah, and not really mushroom, but pathogen fungi and causes. Okay.
I'd like to find out more. It's
Okay.
a hobby of mine.
Oh.
To collect mushrooms. So, yeah.
Yes, thank you very much. Okay, thank you very much for Histology Department for collaboration this event with register program, and I hope it will be useful for us for now and also for our future. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
And last but not least, we are pleased to welcome Dr. Dewi Sukmawati, MKes PhD, Head of Department of Histology FMUI, also the Executive Chairwoman of this event.
Okay, thank you very much, Dr. Priska. So good morning, everyone. Today we are so grateful because we have our new adjunct professor for histology department, but also we can collaborate it with the master program and also doctoral program in biomedical science. So I also very thankful for both of the program and also for all of the staff that help this guest lecture. Yeah, so for today lecture, and I hope I also glad also because today both the secretary also mentioned that this topic will be benefitful for the students. So I mean it is very precious topics, I think. Yeah. So I hope all of the students will enjoy the topic and please ask many questions. Also for the hybrid model, yeah, for the online model, please ask any question and enjoy the lecture. Thank you very much.
Thank you, Dr. Dewi. And now allow me to introduce today's moderator, Professor Dr. Jeanne Adwinata Pawitan, MS PhD. Prof. Jeanne is a professor at Department of Histology, Faculty of Medicine, Universitas Indonesia. She earned her medical and master's degree at FKUI and completed her PhD at Osaka University, Japan. She is an active member of several international organizations such as International Society for Stem Cells Research, the International Society for Cell and Gene Therapy, and the Asia Pacific Cell Therapy Organization. Her research focuses on regenerative medicine, tissue engineering, and mesenchymal stem cell with numerous publications in high-impact journals. Professor Jeanne, please kindly take the lead. Please give applause for Professor Jeanne.
Thank you. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody, students and colleagues. Today, we will have a guest lecture from Otago University, Professor Alex McLellan. Professor Alex, the time is yours.
Thank you so much. Thank you so much for the very warm introduction, and I know you put a lot of work into getting me here. It's harder to prepare the feast and the reception than it is just to visit, so I appreciate all the work that's gone into the adjunct professor appointment from Dr. Radiana, and I appreciate that you turning up here to learn more about immunological cancer models. I hope I don't disappoint or bore. We will get quite heavy into immunology because that is my field. If I could have the first slide, we could begin the lecture.
Sorry, Professor, if I may to present your CV first. Sorry. So Professor McLellan is professor at Department of Microbiology and Immunology, University of Otago, member of International Society for Cell Gene Therapy, Australian Society for Immunology, Center for Translational Cancer Research, University of Otago. And his research activity, his expertise is in immunology, cell biology, and diagnostic development. So I think Prof. McLellan has prepared some presentation material. Okay. So please, the floor is yours.
Thank you once again for the introduction. I'm from Otago University, which is in the bottom of the South Island of two large islands, New Zealand. It takes me a few days to get to Jakarta. I usually fly up to Christchurch, in the middle of the South Island of New Zealand, then to Sydney, and then another 7 hours of a ride to Jakarta. And it's my third time in Jakarta, or third time in Indonesia. This is the first slide. Could you go back to the first slide, please? So this is my first time presenting in this forum, and thank you again for making this available to me and for the warm introductions. So I'm part of the Department of Microbiology and Immunology, and I was hired because I'm an immunologist, not a microbiologist, although I did do a master's on osmoregulation, genetic regulation of salt tolerance in E. coli. And from that I always wanted to move into immunology, so it was a dream realised to be able to do a PhD in immunology with Derek Hart, who was a transplant expert, and we learned a lot about MHC, we learned a lot about antigen presentation and T cells during my thesis in Christchurch at the University of Otago. First slide, next slide please. So I've been asked to talk about cancer models, so I thought I'd focus on what I know best rather than going into what I know little about. Herbal remedies or different cancer models. What we focus on are CAR T cells and CAR NK cells, but I have done a lot of work for the immune response in immunocompetent mice before, so I thought I'd share some of that. So the mouse model is very flexible. There are a lot of different genetic variants of mice available. It can become quite confusing to learn all the different histocompatibility issues that you experience with different mouse strains. Most people might focus just on valve C or C57 black, but there are a lot of different mouse models out there for studying cancer. One of the important mouse models that we use is called NOD-SCID gamma. Why it's called NOD-SCID gamma is it lacks a lot of genes that are involved in the immune system. So it's basically an empty vessel for putting human cells into humanise this mouse model. So that's what we use a lot of. There are also ways to manipulate mice so they can resemble humans. For example, you can knock out the entire immunoglobulin locus for antibody production and replace it with human antibody genes. So the mouse now produces human antibodies in response to immunisation. That's how a lot of new therapeutic drugs are being made to produce monoclonal antibodies using the humanised mouse. This is a major undertaking, and this is not minor allelic replacement. It takes a lot of work and rationale and understanding to be able to do those processes. Companies like Regeneron have done that successfully. In our lab, we take NSG mice and we can transplant them with human PBMC CAR T cells, and we can also transplant them with human tumours. That means that this is a preclinical model because everything is human except for the mouse. So we can study the immune response to tumours using human material, 100% human material, apart from the mouse host. I'd just like to ask you a question that you could think about. You don't have to answer it because I know you'll be quite shy. If you've discovered a biologic that cures cancer, how does it work? Does it interfere with cell division? Does it stop the cancer cell from dividing? So imagine you put a cancer into a mouse and you've treated it with your remedy or drug, and it disappears. The tumour shrinks. But you don't know how that happens. What is the drug doing? Is it acting directly on the tumour? or is it engaging the immune system? In fact, most chemotherapies engage the immune system as well as acting as antibiotics against the tumour. It's very rare that you find that radiation or chemotherapy does not engage the immune system. The immune system will recognise danger signals from a dying cell. Dead or dying cells will activate the immune system. So your herbal remedy may well be acting to activate the immune system through perhaps necrotic pathways, death or dying, or it may be acting in other ways to recruit cells to kill the tumour cell. So in vitro testing might reveal metabolic and cell growth alterations, but a complex animal model is required to uncover the role of the immune system. And if you look at the immune response to a drug, just asked ChatGPT to make this figure for me yesterday. It was a little weird, it was arguing with me, to give me what I wanted. It still hasn't given me what I wanted. But I've moved some labels and imposed my will upon this ChatGPT for this figure here. And if you see there's a drug there, there is undoubtedly an immune response against tumours happening. Of course, some patients will experience spontaneous regression if they're lucky without a drug being present. Tumours are not always invisible. Some have a high mutational burden, which can increase their oncogenic activity, but it can also make them visible to the immune system. How they become visible to the immune system can be through cell death pathways, but to the adaptive immune system, the mutations have created novel peptides, which can be recognised by T cells. So a tumour growing inside an animal might be subject to surveillance by the immune system. T cells might recognise altered peptides on MHC. NK cells will be triggered by the lowering that can happen in tumour cells of MHC1. Sometimes stress ligands can be induced during cell division that will activate NK cells. And macrophages can also be attracted to sites of malignancy and exert an anti-cancer effect. So if your drug is shrinking the tumour, how is it working? So the immune system is a complicated, I hope this is the next slide. The immune system is a complicated network of cells that cooperate. And so when you try to uncover individual cell contributions, you must be aware that the immune system acts as an orchestra. Every cell helps every other cell. This nearly always happens, sometimes very directly. For example, the role of CD4 helper cells is well established to help other cells. So cooperation is essential between immune cells, and sometimes you might find you can knock out several cells and they will all affect the tumour, anti-tumour response. NK cells are newly discovered helper cells. They can actually act similar to CD4 cells and assist CD8 cells kill tumour cells, something we showed a while back. Helper cells, of course, have their well-established roles in helping CD8 T cells become cytotoxic against cancer cells, and sometimes the B cells can be active in producing antibodies which can target the tumour. At the start of it is the cell. I did my PhD on the dendritic cell, which is a powerful antigen-presenting cell. And a while back, dendritic cells were going to cure cancer, so it was thought. But then conference after conference after conference, people were presenting more and more depressing results. The dendritic cells loaded with peptide didn't cure cancer, or loaded with cancer antigens. The problem was, of course, that the T cells were becoming exhausted and falling over, and it wasn't enough just to keep offering them food back here. They needed some sustenance in their journey. And that's why a lot of the cancer immunotherapy is now focusing on the adaptive cells, the T cells themselves, to try and make it persist longer in the cancer patient. So NK cells are a favourite cell of mine. I think they have an important role to play in the anti-cancer response. And we uncovered this in a number of studies shown here, that they are essential for dendritic cell-based immunotherapy against melanoma. This is where the dendritic cells are loaded with peptides specific for T cells. But the NK cells are nevertheless essential for this response. And you can see here that we uncovered that the NK cell will help an anti-cancer response at the start, at the priming phase. And the reviewer asked us during the submission of the manuscript to have a look at the effector phase, when the tumour was introduced into the animal. Were the NK cells important to help T cells? And the answer was yes, which was very surprising. So NK cells are present at the priming stage and exert activity there, and they're also required at the so-called effector stage when the CTLs are attacking the tumour. They encourage the CTLs to attack the tumour. So NK cells help in an interferon gamma dependent manner to assist the killing, but also the earlier events of priming. So the question you might want to ask if you see a response and you are interested in the immunology behind a cancer response, and you want to uncover which cells might be responsible. It's difficult. Cancers do not induce strong immune responses like virus infections do. CD8 T cells can be induced to become proliferative, highly proliferative, and make up 5% of the T cell population. But this is during a virus infection or during some acute bacterial infections like listeria. You get CD8s high. With cancer responses, you won't see the T cells induced to very high levels. Likewise, a meaningful response with a CD4 cell, you still don't see many. This is hard to monitor. And B cells, of course, these are really hard to uncover. You can use a technique called an ELISpot, which is quite good because it's a re-stimulation protocol in vitro. But generally, those sort of numbers are hard to look. It's very challenging by flow cytometry, even using tetramer technology, which you can use, becomes a challenge. So how do you uncover those? Well, one of my favourite methods is to use monoclonal antibodies to simply deplete those cells before the drug is introduced. Or shortly after.
Dia harus di itu, Bu.
Or polyclonal antibodies can be used to deplete those subsets that you assume might be important. If you are listening and you are unmuted, could you please mute because there's some interference coming through for the lecture. So we'll have to look to see who's unmuted in a minute.
It's okay.
Okay, thank you. So monoclonal and polyclonal antibodies can be purchased, they're commercially available, or you can make them in-house. And they're usually used to deplete those important subsets of CD4, CD8, or CD20, which are used to deplete the B cells. Dendritic cells are harder to deplete, but you usually use a mouse system that has a diphtheria toxin receptor engineered in that can deplete specific subsets of dendritic cells, not the new This set of tools here would allow you to uncover which cells are important in the immune response to cancer. And this is an example from one of our publications where we've done that, and this is an interesting finding here because we've immunised with dendritic cells and we've looked for the immune response. So we've just immunised the saline there, no response. We're looking at percent of killing of a peptide pulse target, so it's an MHC1 dependent immune response. With the mock depletion here, we're using a control antibody. This is the cytotoxicity here, quite high, about 50% of the targets that are injected are killed. But this is interesting, it shows the cooperation. If you deplete CD4s, CD8s, and surprisingly NK cells, you see a drop in the immune response. So this is an example of the cooperation that is happening between cells and combating the tumour in response to an immunisation step. B cells, not specifically required for this. This was a CD20 depletion. You can produce the antibodies in-house. Perhaps we could help with supplying plasmids for doing that. Or we also recommend this company here. They're not too expensive for buying monoclonal antibodies to use for in vivo studies. Do you want to read more about that technique? It's in this Saunderson et al. 2017. Next slide, please. This is an example of using antibodies to deplete soluble factors, which is again a fairly simple experiment to carry out. It's similar to depleting the cells, but you're neutralizing a cytokine. In this case, it was interferon gamma, and we got lucky because in this immunization here, you can see the effect here with the dendritic cells inducing an immune response against melanoma. This is melanoma growth here. It's been reduced by the immunization. That is lost when you treat with an anti-interferon antibody there. So it's using antibodies to uncover a pathway. Down below, we've used perforin knockout mice, interferon gamma knockout mice. You can actually see that the interferon is more important in many solid cancer models. It's the loss of protection here in the interferon knockout dendritic cell treated mice. Interferon is more important than perforin granzyme for many of those anti-cancer responses, which does surprise some people. Now, one convenient way you can look further at T-cell responses is to look at cell proliferation. And this is a common technique used, you might find this in many journals that you read, using a cell proliferation dye. It's a simple fluorescent dye that can be used to label lymphocytes, and you can track their cell division, because as they divide, they lose half the fluorescence in each cell division. How many of you are familiar with this technique? Okay, it'll be a good one to explain. Even if you don't use it, it's quite nice to understand if someone's talking about it in a manuscript. So what you get at the end is you see these are undivided T cells, and these are divided T cells. At each stage of cell division, they've lost half the dye, and so you can trace their proliferation. You can also gate those cells and have a look at the immune markers on them, see how T cells change. It's a very valuable technique. And this is an example here where we've used it to trace these T cells here, which have extensively proliferated after a few days. You can see they've lost all the dye. These are control animals that were not immunised, where the T cells have kept all their dye. You can track T cell proliferation in response to an immunisation or perhaps other drug treatment. In the mouse model, to use the CFSE method, we have often used transgenic mice as the donor mice. The transgenic mice are OT1 or OT2. That means they have a high precursor frequency for over-specific T cells. They're engineered by Bill Heath in Melbourne, and they are knock-ins, so they have a T cell receptor that recognises a certain peptide. So every T cell you take out of that mouse that is CD8 or CD4 will have those receptors on, and so you can trace them, get a very strong response. The problems here, I don't really like this anymore because the higher precursor frequency can result in non-physiologic immune responses. It will overcome requirements for co-stimulation. And I think you'll find that the OT1 and OT2 models are being used less and less frequently these days, but that is one way they have been used in the past. You can still use the CFSE on, for example, peripheral blood mononuclear cells. You can label them, and you can still trace T cell proliferation in vitro. So I've left this method in there because it's a very convenient method for measuring T cell proliferation in vitro in response to anything. A re-stimulus with a peptide, a re-stimulation with a protein, and perhaps a parasite protein. You can trace cell division using CFSE dye. Don't be tempted to use another dye. Often they don't work. CFSE is a little unusual, and there are some substitute dyes you can use in violet or red colours which will also work. But don't just grab any dye, not all from work. Right now, into the 21st century, bioimaging. So this has revolutionised the way we look at tumours. So if you want to study a tumour, perhaps you've made a primary tumour line from a patient, or you have received a tumour line from the ATCC. What you first need to do is make it luciferase positive or luciferase expressing, so that you can have a look at how it's grown in vitro or in vivo. So what we use is luciferase gene, which is very easy to insert using the transposon system into the tumour cells. So like a one-step transfection, if you like, using two plasmids, simple transposon system. All of the tumour will express that marker, the luciferase, and you can drug select for it. You get very high luciferase expression. It's high throughput and cheap. If you're doing 96-well plate assays in vitro using adherent cell lines, you can run through these in just a few hours. Often it's an overnight incubation, so you can go home and sleep. Come back the next day and add after a wash, just add the luciferin substrate. You can see cytotoxicity as a reduction in luciferase activity. Very nice way of measuring anti-tumour responses. The tumour cells, once they're luciferase labelled, or now they're expressing luciferase because the luciferase gene is in their genome, can be imaged with a bioimager. These examples of mice that have been implanted. This machine here is the same one we've got in Otago. It's very nice because it's got an X-ray component in. I bet we don't use the X-ray much, but we could. And with the X-ray, particularly for metastatic cancers, which we're not studying much of at the moment, the X-ray gives you nice contrast in relation to where the bones of the mouse are. But what we're mainly interested in is just looking at the intensity, the flux of this luciferase expressing tumour as we treat the mice. We image the mice, anaesthetise them, put them in a bioimager once a week. That's enough. We get very accurate data from that kind of study. Measuring cytotoxicity, if you want other alternate methods, not using luciferase, you can always just label your tumour cells with a dye, for example, CFC, pop them in with the effector cells, and then you use a flow cytometry setting to determine killing. Do not make the common mistake of doing a set event acquisition requiring 20,000 events. Why? Because when you collect the cells and you wash them, all the cells that have been killed explode. They're lost in the saline washes that you're doing before you put it through the machine. So always do a set volume acquisition and calculate against the control. Otherwise you've underestimated greatly the percent killing. So always use a set volume. So you tell the flow cytometer to require 100 microliters. Don't tell the flow cytometer to require 20,000 cells or whatever. Always set volume. Otherwise you underestimate the killing due to cell losses, dead cell losses during cell processing. Very nice, very sensitive method, and it can just be done with the flow cytometer. I can make these slides available to all of you if you like, if you don't want to be scribbling down notes. The in vivo cytotoxicity assays using CFSE might be used for peptide-specific killing. An example there would be taking peptide pulse splenocytes in one tube and unpulse in the other tube and giving them different doses of CFSE. Then you can mix them together and you can identify them after flow cytometry. You've got internal control, and that's good because it minimises variation. You can also inject these into mice, and after 24 hours after IV injection, the ratio of high to low CFSE expressing targets can be determined. And it's also possible to use mixtures of cells expressing violet or far red dyes. You can go up to 4 or more conditions using different peptides, different cancer peptides and so forth. So the dyes that I've been talking about are good alternatives to luciferase, and you can use them for many in vitro assays, but you can also use them for in vivo assays. And this is an example here. We've used a very simple setting. We've pulsed half the splenocytes that we're using as targets with a peptide, and half we've left unpulsed. Now those two preparations we given different doses of CFEC, so we can later just gate them on the flow cytometer. Very easy, very cheap, and very simple to run. And this was, again from this, I've already shown this video here, this was actually performed using ratios of high CFEC to low CFEC. And so those high CFEC would have had the peptide, low CFEC had no peptide. So you just determine the ratios. CTLs will not kill the unpulsed targets. Those are the internal controls that you've always got. This is a very nice system to run. When you're looking at animal models, we need to be aware of the confounding effects that you get if you start mixing species together, or mixing strains from an outbred strain. Some rats are outbred, some mice are outbred. Some are pure inbred, such as Bell C or C57, so they're syngenic. Usually no little problem between sharing material between those. But if you start going mouse to human, or human to mouse, then you get a xenogenetic response. And you have to be aware that this will be occurring. And it's about 5% of T cells that are xenoreactive or alloreactive. That means for a random donor in an outbred population or between species, about 5% of T cells can react to the tissue from another host, another donor. Examples here, if a human tumour in mouse is xenogenic, even if the MHC1 negative cells are used, or MHC2 negative, it will still be able to induce a xenogenic response because of the NK cells. The NK cells will recognise the loss of MHC and they will miss certain inhibitory receptors, and so the NK cells will become activated against the other species. Even male to female might present a problem if you're using a mixture of male and female inbred mice. There is a measurable male to female immune response in mice. If you inject targets or prime a mouse with male splenocytes, you can detect that the female will produce T cells that will kill the male tissue. Sometimes it needs a re-stimulation to visualise, but it's definitely there. So you have to watch out for allogeneic, xenogenic, even male to female responses for these assays. Sometimes the minor histocompatibility issue might be with another protein other than the HY, but it is an interesting example here. Even in the same strain of mouse, male to female can give an immune response. And that is why we have polymorphic MHC is obviously to present a great variety of peptides. So MHC between different individuals will have different mutations. They will greatly vary. You nearly always see an immune response in an outbred population of one species. And between species, you'll also see similar strong responses between MHC and alloreactive T cells. And NK cells are also involved in that alloreactive response or xenogenic response. But NK cells also react or bind into MHC. They actually bind into an overlapping position in the peptide binding groove. These are the killer inhibitory receptors, bind to MHC. So that's different between host and donor. You can still get an NK cell response. And an important thing there is there's little inhibitory or little shared inhibitory signaling between mouse and human NK cell ligands. That means, for example, putting human tissue into a mouse, you will trigger the mouse NK cells. They won't see the inhibitory receptors because they're not there due to evolution. So NK cells are important to match or to think about your response when you're looking at the confounding features of treating tumours with your extract. And they will respond to missing self. So, have I got about some minutes left, or how much longer have I got?
About 15 to 20, but it's okay. You can continue.
Okay, I'll get through. Yeah, have faith. Okay, so I'll just talk quickly about something that we doing pretty much all the time in the lab now. Some of the techniques I don't think we will use very frequently anymore. We still use the CFSC method occasionally, and definitely use luciferase, but I want to talk about adoptive cell therapy, which is a way to cure cancer, and it can cure cancer in a number of patients. There is still a treatment gap with CAR T-cell therapy, although it can be used to treat patients that have failed conventional therapy. So it is a new hope to expand into solid cancers. So there are two types of adoptive cell therapy. One where you can get the T cells from the tumour itself, which is called tumour infiltrating lymphocyte therapy, or you can take the cells, usually from blood, sometimes from cord blood or allogeneic sources, grow them up in vitro bioreactors, and then you can either engineer them with a CAR or put them in just, if they're from, especially from this setting here, they will usually be reactive against the cancer. They just need a bit of encouragement here. Put those back, and hopefully the end result is tumour destruction. That is adoptive cell therapy, which includes this new therapy, which has been FDA approved. So this is the FDA approved TIL therapy. Lifalusil is now an FDA approved adoptive cell therapy developed by Stephen Rosenberg here. This is an early result here showing metastatic melanoma shrinking in response to unmodified T cells that have been taken out of the tumour. They were exhausted in the tumour, no doubt. Melanoma commonly has quite a lot of infiltrating T cells. If you're a histologist and looking at melanoma, you nearly always see T cells in the melanoma lesions. However, those T cells you can reawaken in vitro by giving them lots of yummy cytokines. Interleukin-2 is one that you give them. As soon as they start dividing, they forget that they were in that exhausted immunosuppressive environment. They start to come alive, and you've got masses of T cells to be able to put back into the patient. So it's a major bioreactor undertaking, quite expensive procedure, but it can show remarkable results. And what we are working on is not lifalusil, but CAR T cells, and this is where you engineer T cells after you take them out of the patient. You activate them to introduce an lentivirus that has the CAR inside the lentivirus and integrates into the genome of T cells. This has shown remarkable efficacy against B cell lymphomas. B cells have an antigen called CD19, and CAR T cells are able to wipe out the malignant cells using this anti-CD19 CAR. Unfortunately, CAR T cells will also take out the non-malignant B cells that have CD19. The patients are treated with CD19, CAR T cells no longer have normal B cells, and hopefully no malignant cells. And it's effective against relapsed and refractory diseases, which shows you that the mechanism of killing acts through pathways other than antibiotic chemotherapy. So it's a new way of killing, if you like. It was an old way of killing, it's become a new way of killing if you can harness it. So the cells, or the cells that we use as CAR T cells are derived from peripheral blood, and we need a preclinical model to study those. We use again the mouse system, we use the NOD-SCID gamma line, which we got from Jackson Laboratories. There's a few varieties of this available. The one we have is quite immunodeficient. What this allows is transplants to be carried out from human material without any rejection. They lack lymphocytes, they cannot reject human cells, they cannot reject the CAR T cells, they cannot reject this tumour on their own. The NSG are vessels for human cells. They can be humanised, you can put in PBMC, you can even incorporate some monocytes into that inoculum or that bolus so that they have a more normal myeloid system for the time of the experiment. And one drawback of NSG is at late time points, 60 days post T-cell implantation, you do see some graft versus host disease. Can anyone tell me why? Why would you see graft versus host? It's an immunodeficient line. Putting in CAR T cells and PBMCs. So this is graft versus host. a question for the students. Okay, after a time, the T cells will recognise the MHC in the mouse. The MHC is still present in the mouse, so it's a xenogenetic reaction. You do see graft versus host. Fortunately for us, it takes about 60 days to materialise. You can see some ocular involvement. Sometimes the skin becomes, like a scurvy mouse becomes ruffled, and the mice have to be sacrificed. So you cannot do long-term, several month experiments. You can do a month and a half about, but longer than that, the NSG model has a weakness, and that is caused by the graft versus host disease. This is an example here of an experiment we carried out where we were comparing different conditions, and you can see that we image every week. You set up an experiment so they have almost equal amounts of tumour present in the flank of the mouse. It's a breast cancer model. Treat with the CAR T cells about 10 days later, so it's a well-established tumour, and you can track the activity of the CAR by looking at the tumour disappear. These mice are actually cured. This one here is not so cured. And in the PBS group, when you see these pictures, you see that mice have disappeared. Obviously, they've been removed from the study because we can humanely remove them by telling our operators to remove them with a certain tumour flux. When the luciferase reaches a certain total flux, we can remove them from the study. We don't wait for mice to die. That's a very humane method to use. So that's an example of a bioimager experiment, just simply imaging the mice every week. The details behind that I'll talk more about Monday, because that was comparing two different promoters in an attempt to reduce T cell exhaustion. And you can see here that our promoter here, this NFAT-based promoter for driving the car, seemed to be the winner. Sorry, it's NR4A1 here, seemed to be the winner. It lost the mouse. This was our promoter here. What we usually do is use then GraphPad Prism to plot the results, and we use a Kaplan-Meier survival curve so we can determine the tumour flux over time and the survival, which is probably the most important data here, is the survival curve, where most of the it's a very precise way of determining the efficacy of a product. If you don't want the mice to die, I don't know what the regulations are for in Indonesia for handling mice, but in New Zealand we certainly don't want mice to be killed by the tumour or the treatment, so we try to develop humane endpoints, which is the total flux of the tumour. So there's two measurements you can get there. There's the flux of the tumour there, and you can see the untreated cells, untreated mice, and the tumours have taken off, plateaued. We've removed some mice, that's why it's gone down. It hasn't gone down. It's because we've been removing mice from those groups. You can see some of those were treated. Some of those mice were actually cured there. It's represented by the survival curve there, where 100% of the mice in at least one group were surviving by the end. Thanks to the lab group, this is a small selection of the people in the lab. And this, do you have April Fool's Day here? We play tricks on other people, April Fool's. We do in New Zealand and it's an English thing too. April Fool's Day, usually there's a fake newspaper article on they're going to build a stadium in the middle of Dunedin or something. People get fooled and they start writing a letter. So on April the 1st, the students dressed like me. This is often how I dress like this guy here. Jeans, running shoes and a shirt, and they also dressed like me as a joke. And I didn't get it. I thought I was complimenting them on the shirt. I told Tom, that's a really nice shirt you're wearing. I was laughing. I like playing tricks with professors. I hope they do it here. So this is a lovely group that does so much work for us, and I'd like to thank them, and in particular the new intake of students here, the honours crowd, master student, and the wonderful technician Thaloma. Also, I've missed off there, I can see Kate P is the new postdoctoral fellow. We have a number of support collaborations throughout the world, including Melbourne, Bangkok, and of course in Indonesia we have some wonderful people working together with us, and we have some CAR T-cell collaboration. We hope one day that CAR T-cells will be available for Indonesian patients together with CAR NK cells. So leave it there. And again, I thank the audience for their patience during my very technique-rich talk. I hope it wasn't boring for you. And thank you again for the invitation to speak.
Thank you very much, Professor Alex. Very interesting lecture. Yeah, in brief, Prof. Alex has explained us how to make an animal model that should mimic human system. Yeah. And then by knocking out the system, our system, and then knocking in the human system, and how to make a tumor animal model by using SOD seed mice and transplant the tumor cells, and how to track the tumor cells, for instance, using luciferase X cells that can be traced using a bioimaging system, or by tracking a dye to the cell and then trace the cells using flow cytometry. I think that is in brief that has been explained by Professor Alex. Professor Alex also explained about the immune system using the CAR T cells, NK cells, and tumor infiltrating lymphocytes. Thank you, Prof. Alex, for the very interesting lecture. Now for the audience, are there any questions?
From Zoom.
Oh, there is one question from Zoom. Bisa ditanya?
Mungkin bisa minta mulai bicara setelah kita mute kita biar nggak feedback.
Okay. For Zoom audience, we will mute first, and then you can ask directly.
Untuk Nurul, Ibu Nurul Gusti, untuk Dr. Nurul, silakan.
Thank you very much for the opportunity, Prof. Jeanne, and also Prof. McLellan. That is such a very inspiring presentation. My name is Nur Gustotima. I'm a PhD student here in Universitas Indonesia. And I want to ask two questions. For the first questions, among mice, rabbits, or rats that are usually used for animal models, which species is more representable for cancer models, which could be similar to human? And the second question is, are there any challenges in inducing cancer in brain or brain tumor in animal models? As we know that all vertebrates have a blood-brain barrier. Thank you very much, Prof.
For using animal models is how well established is the model and how many tools are available. For example, there are many more antibodies available for analysing mouse tissue compared to rabbit. And I think mice will win over rats in that regard, but that doesn't rule out using those animals at all. It's just at Otago we're more used to working with mice. They're cheaper. They breed well, and even the NSG breed very well. So they're well facilitated animal model and thank you, nice to sit down. And with the NSG model, which is a severely immunodeficient line, it can be implanted with human cells, so it brings you a little bit closer to humans. But in terms of evolutionary similarity of the receptors, rat, rabbit, mouse, possibly they've all got their advantages and disadvantages in terms of evolutionary distance. That's as a drift apart from humans during evolution. So there's no real model that is exactly like a human. Of course, the closest are primates. We cannot do primate research in New Zealand. Probably a good thing. I wouldn't like working with monkeys. They look too much like me and probably feel too much. The mice are for us the ideal model. There might have been a second part to your question. Brain tumours, yes. Yes. So the blood-brain barrier can be overcome by IV injection. So T cells will surprisingly bypass that, particularly if there's a little bit of inflammation. But intracranial administration of CAR T cells is now being carried out in humans, and this is highly repetitive treatment. So some kids have had glioblastoma that have had multiple CAR T cell treatments, and they seem to be holding back the tumour growth in some of those very young patients. So often, to bypass it, you put the CAR T cells directly on the tumour.
Okay, thank you very much, Prof. McLellan, for the answer.
Boleh ditanya feedbacknya, Dr. Nurul, ada
Any
feedback?
more question, Dr. Nurul?
I've already get all the informations. Thank you very much, Prof. Jeanne. Thank you very much, Prof. McLellan.
Maaf dok, tadi ke mute, boleh di...
You are muted.
Yeah, I already understand, Professor. Thank you very much.
Any more question, Dr. Nurul?
No more questions, Prof. Thank you very much.
Okay.
It's all clear, Prof. That's it. Thank you.
Okay, from other audience in the Zoom, are there any questions? If there are no more questions from the Zoom, now from floor, are there any questions? Yeah, Fuad.
Thank you for. Okay, my name is Prof. You can call me Prof. So, thank you. I want to ask about the basic question about the method we use to develop the cancer model. So, in the cancer model, we only have the tumor microenvironment, and if you want to test about the ability of the immune cell, even the CAR, about the ability to eliminate cancer, which may probably be the good one from your opinion or your experience to develop the cancer model. Is that the humanized model or by the chemical model? If we consider about the tumor microenvironment.
Yeah, so using an autochthonous genetically predisposed model or chemically induced model has an advantage of, if you're doing just strictly mouse studies, mouse tumour, obviously they're endogenous, they're not grown in cell culture, they have quite an advantage over implanting cell lines that have been multi-passaged. So there are advantages of both ways. Obviously it's harder to chemically induce tumours as a more difficult, more expensive model, and it depends on sometimes the models have to be predisposed, lacking certain tumour suppressor genes to enable that to happen. Sometimes they can happen without tumour suppression. Is there another?
Maybe yes. Yeah, but so the good method for the testing was using the humanized to implant. This is Prof.
I have limited experience with autochthonous or chemically induced tumour models, so I shouldn't comment on that. But what we need for CAR T cells usually is just a humanised mouse system, and then it's convenient for us to inject the tumour, watch it grow, and put in the CAR T cells. So let the tumours establish. The disadvantage of our model is that the tumour microenvironment is murine, so it's difficult to target or to develop strategies to target that unless you engineer the mouse further. that's a disadvantage of that model.
Okay, thank you for your answering, Prof. And it's answering many questions, answering curiosity for me.
Thank you.
Okay, thank you, Prof. Alex. Any more questions from the floor? Yeah, what's your name? I'm sorry? Yeah, Husnul. You can ask.
Hello, Professor McLellan. My name is Usno Azahra. I'm currently studying microbiology in master program in biomedical science. And I'm very interested in host microbe interactions and immune modulation. So I found your research on CAR-NK therapies in solid tumors. And I was wondering, since the tumor microenvironment tends to be immunosuppressive, have you considered combining CAR-based therapies with microbiome modulation or microbial adjuvants to enhance in vivo immune responses? Thank you.
Thank you for your question. It would be really interesting to look at directly targeting bacteria to tumours. There is some work in that space happening. Modulating the microbiome is not something we've looked at, but we're trying to follow the system because some patients have a poor microbiome that translates to worse outcomes with some cancers. And there's a tumour microbiome that exists, and there are some bacteria within that tumour, I forget the names of them, that predispose to a worse outcome. So the microbiome is important not just for the gut and metabolites that generate, but also within the tumour, the colorectal tumours. But it would be fascinating to combine some directly targeted bacteria that are home to the tumour to assist the NK cells and the CAR T cells. Thanks for the question.
Maybe another question from students.
Dari peserta yang ada di Zoom, apakah ada yang mau bertanya lagi?
Peserta di Zoom, silakan kalau ada yang mau bertanya. Any more questions from the Zoom audience? No? Maybe from the students? Any more questions?
Can I just finish answering that question before, because Indonesia has a company called Universitas, which look at microbiome makeup, and perhaps I'm not sure whether you engage with them, but that would be an interesting company to perhaps track CAR T cell responses by looking at the corresponding microbiome. Have you come across Universitas? A company, an Indonesian company in Jakarta called Universitas. They actually look at microbiome composition by genetic means.
Pardon me?
Universitas.
Index for microbiome.
It's a company in Jakarta that look at microbiomes by genetic means. I wonder if you come across those heard of them. You can Google them, believe me, I'm not making it up.
If the student no question, I will ask. You, Prof. Actually, I have two questions. First, in Indonesia, there are many research about cancer, especially about to test herbs from our country which have anti-cancer activity or not in animal model. Have you suggestion which maybe animal model that's suitable for maybe like doing quick testing or quick assay to test which is whether the herbal has anti-cancer activity or not? That is the first question. And then the second question is, I'm very interested in TIL, tumor infiltrating lymphocyte. I saw in the picture that the lymphocyte is extract from the patient and then doing like culture and then treatment and then going back to the patient again, IV injection again. I just suspicious whether is it possible that the lymphocyte should be from its own patient or it can be from another patient. How is the possibility or how is the practice in your hospital and maybe in university? Thank you.
So the first question is about a mouse model you use.
Yes.
First question, which mouse model might be appropriate for testing extracts against cancer? Probably the simplest is an inbred or even inbred mouse strain with a well-established cell line that is of the same genotype. It's probably the simplest way. You don't need to use humanised at this stage. It's simple and cheap, and you can obtain those cell lines relatively easily. Pancreatic cell line, for example, if we're looking at pancreatic cancer, they can usually be implanted ectopically for easy monitoring, and you can still monitor immune responses against or extract responses, chemical responses against the tumour using that model. Then if you see a response, you could get a bit more complex in your model system. Perhaps look at NSG mice and introducing human cells or using human cancers in the NSG system, which is an advantage. And the question about TILs is, the TIL therapy is one of the few that you can't use allogeneic. It relies on T-cell recognition of MHC cancer peptides, so it has to be matched. It could be from a twin, possibly even haploidentical, but usually it's autologous. So the tumours, and also because it's tumour related, the cells are taken from the tumour the patient has. But generally that sort of therapy where the T cells have been stimulated to an extent by the tumour peptides are introduced back, has to be autologous. CAR T cells can be allogeneic, but there is a rejection that will occur. But the rejection of cells in CAR T cell patients is minimised because CAR T cell patients are heavily preconditioned with cyclophosphamide, fludarabine, before treatment to enable the CAR T cells, once they're introduced, to rapidly divide and fill the empty space. So that's one way you can introduce allogeneic cells. But there's always a risk of GVHD, there's always a risk of the cells getting rejected.
Okay, thank you, Prof.
Okay, any more questions from the audience? Both from the Zoom. If there are no more questions, I will have a question. May I?
Yes.
Okay. I am intrigued by the questions before from chemical animal models. Yeah. If we are to test CAR T cells, for instance, it is human cells, right? And if we test it in an animal model, chemical animal model like rats, yeah, maybe the cells, as you have mentioned before, the cells can be attacked by the immune cells of the rat, right? So, if Mr. Fuad want to use chemically induce a tumor in an animal, should he use CAR T also from rat? or can he use from the human?
Human would likely be rejected. Rat could still be rejected if the strain is an outbreed strain. But it depends on some mice and rats are outbreed and some colonies are inbred, meaning they're pure for the MHC expression. So it does depend on the strain used, but generally, if it's a xenogenetic transplant, meaning human cells into a rat or a mouse, you'll expect a two-way reaction. You expect rejection of the cells and possibly even graft versus host disease. That can occur for the cells you're using to treat. It can also occur if the tumour itself is not matched to the host. So mismatching can occur in between outbred rats as well. So it's complicated.
Okay, thank you. And you have also mentioned before about humanize animal by knocking out and knocking in. What do you mean actually by knocking out? What is knocking out? The cells, only a certain cell of the animal is knocked out, or the whole animal is knocked out. So that means you knock out the embryo. You knock out the embryo.
Yes, so usually those mice are generated using ES stem cells and blastocyst implantation, and usually some form of allelic replacement, which can be CRISPR-Cas assisted, or more traditional methods of homology-directed replacement. So in those mouse lines that have human immunoglobulin, they replaced the mouse immunoglobulin locus in the genome of the ES cells with the human immunoglobulin genes, so that when they implanted those into a pseudo-pregnant female, then they could generate this new line of mice through breeding that has no mouse antibodies, but it produces only human antibodies. So 100% of the offspring after selection should produce just human antibodies. The rest of it looks like a mouse. Everything else in that mouse is mouse-like, except for the antibody genes. And they can get quite complicated, those studies, if you read some of the Regeneron, which is a large company that's generated those mice. One of at least two companies generate those mice. Regeneron might be doing a study where they're looking for a new bispecific antibody, and they will quite happily in a few months produce another knock-in that has human CD28 or human CD3 knocked in. Some companies are just very fast at producing these humanized mice, which is done by allelic replacement, so it's a permanent integration into the gene. Once you've done the selection of the mouse through outcrossing, then you can select for those homozygotes that have the human insert.
Okay, thank you very much for your answer. It's very interesting. But again, you will need a genetic engineering, right?
It depends. You can buy some of those mice from GC,
Yeah,
so some of them are available.
Yeah, okay, thank you very much. Any more questions from the audience? So if you want to make humanized mice, you have to use genetic engineering.
Hello, thank you for the opportunity. My name is Jennifer. Maybe just a little bit follow up about the thing that you mentioned before. So when we create the animal models with the knock-in and knock-out, how do you make sure that this is not integrating with the result? Because we do something with the mice, right? How we know that the knock-in or the knockdown is not really interfering with the process of the body to responding to our the things that we do to the mice. Thank you.
Yeah. So. Thank you for your question. So there's two types of humanization I talked about. One is using just cells, which is what we do for NSG mice. We put the cells in, and we call them humanized. There's the genetic technique that has been developed, not by our lab, which is to insert some genes and knock out, at the same time you replace the murine ones. And those studies this is a really good example, and those studies that replace the murine antibody genes with human antibody genes, it was quite an undertaking. They had to turn around one locus, reverse another one, reverse some promoters to make sure that some of the B cell activation factors that are nearby on the chromosome were also working or replaced by human. So in terms of B cell responses, they tried to make those mice have as human as possible B cells. Those mice are tolerant against those new sequences, so the pharmac selection will make sure the T cells are tolerant against those foreign sequences that have come in. A developing mouse usually is very tolerant to all those new structures that are inside it. So there are very little problems experienced with those mice as models, but those mice are often just used as tools to generate antibodies. They're not always used to study the immune response to antibody. They're often just used as a tool. If you immunise those mice, you automatically get a human antibody without even needing the sequence. You can guarantee all your antibodies are 100% human, because if you have a drug that is antibody-based to use later in a patient, it must be human or humanised. Otherwise, you get an anti-drug reaction. If you put a mouse antibody into me to try and cure my cancer, eventually I'll reject that antibody because it's mouse. It's got a different sequence. So those mice overcome that problem. And they make sure you get quite a variety of antibodies produced from a single immunisation strategy.
Okay, thank you, Prof. Alex. Any more questions from the audience? From the Zoom audience, any more questions? Okay, if there are no more questions, I will give back to the Master of Ceremony.
Thank you, Prof. Alex and Prof. Jeanne, for this interesting session and discussion. Next is we would like to express our gratitude to Prof. Alex by giving a token of appreciation or maybe certificate. Therefore, we invite Dr. Dewi to give a certificate for our speakers.
Cek dulu isinya.
Okay, yeah. And maybe, Prof. Alex, would you please stand up and receive our token of appreciation? Boleh minta? Yeah, please show it to the camera.
Iya.
One, two, three. Thank you. Maybe we want to invite all the lecturers and staff of FKUI. First, to take picture with Prof. Alex and lecturer and staff first. And then later we will do with all the participants. Wait a moment.
Udah sama mahasiswa. Udah lewat sini. Mahasiswa dulu Pak. Mahasiswa.
Maybe we can say stem cells, cancer cells. Okay, thank you. Oh, okay. One, two, three. Maybe you want to take another pose? Really uncomfortable. Okay. Before we end this, Burani, would you please to give the certificate to our moderator, Professor Jeanne? Ya, boleh foto. Oke, foto ini untuk Prof. Jeanne. Okay. Next, we will take the picture. When you actually please wait for when we want to take a picture with the participants. Oh, okay. Just one. Prof, please. Prof. Jeanne. Gimana caranya saya mencari ya? Is there any Zoom operator that can press the screenshot button on button? All the participants in Zoom, you may open your camera so you can take pictures together. Good. Duplikat aja nggak usah. Nah, screenshot di sini. Okay, we will capture it from the laptop. Bisa jadi satu aja, kan dua di scan. We're adjusting the camera so it can fit all of us. Ya udah begini aja. Kecil. Okay. Ready, everyone? Okay. 3, 2,
Cheese. Saus-saus.
Next page ada.
Not all the participants Yeah, thank you. Okay, so thank you everyone for joining our guest lecture today. We apologize for any shortcoming in this event and have a wonderful day. See you at next academic event in FMUI. Thank you.
Dulu panas dulu, bagus. Iya, tapi ini stop aja nanti kami
Iya. Mungkin aja udah.
Because they work together, they support each other.
Iya. Ada itu nih.